This video will cover the citric acid cycle. This cycle occurs in the matrix of the mitochondria. The TCA cycle uses oxidized glucose in the form of two 3-carbon structures known as pyruvate. To transport pyruvate into the mitochondria, a pyruvate proton symport moves pyruvate into the matrix. Once it is in the matrix, it is converted to acetyl-CoA through the complex called the pyruvate dehydrogenase multi-enzyme complex, or PDH for short. The PDH complex is made up of 3 coenzymes: pyruvate dehydrogenase, dihydropoyl transacetylase, and dihydropoyl dehydrogenase. Along with this, NAD+ and CoA are also involved in this conversion. The PDH complex requires CoA and NAD+ and produces NADH and H+, which is passed into the mitochondrial membrane space. CO2 and acetyl-CoA stay in the matrix. In the first step, a condensation step, acetyl-CoA is combined with the 4-carbon oxaloacetate molecule, forming the 6-carbon molecule citrate. The CoA release stays in the matrix. The enzyme utilized in this reaction is Citrate Synthase. This step is highly exergonic with a delta G of negative 32.2 kilojoules per mole and controlled by ATP through negative feedback. Both NADH and Succinyl-CoA are inhibitors of the enzyme. The equilibrium of this reaction favors the products, therefore making this step considered to be the committed step of the TCA cycle, thus making it highly regulated. The next reaction involves isomerization of citrate to isocitrate using the enzyme aconitase. In this reaction, citrate undergoes a dehydration, then rehydration, converting citrate into its isomer. The reaction has a positive delta G, 13.3 kilojoule per mole, and the equilibrium lies to the left. Even though this reaction is endergonic, it can be preceded by the next reaction being highly exergonic and coupling the reaction and pulling the reaction along. In the third step, the reaction involves the conversion of isocitrate to 2-oxoglutarate, also called alpha-ketoglutarate. Isocitrate is oxidized and produces a 5-carbon molecule through the enzyme isocitrate dehydrogenase. NAD+ is reduced to NADH, and this is the first NADH producing step. Along with NADH, CO2 is also produced from the loss of the central carboxyl group. High levels of ATP and NADH will inhibit the isocitrate dehydrogenase enzyme. This is a highly exergonic reaction with a delta G of negative 20.9 kilojoules per mole. Step 4 is another highly exergonic reaction with a delta G of negative 33.5 kilojoules per mole. This reaction involves the conversion of 2-oxoglutarate to Succinyl-CoA. The enzyme complex is oxoglutarate dehydrogenase complex, a multi-enzyme complex like pyruvate dehydrogenase complex. The 4-carbon molecule then binds with coenzyme A and forms Succinyl-CoA. In the oxidation, NAD+ is reduced to NADH and CO2 is released.
Following step 4, the Succinyl-CoA is converted to succinate using the Succinyl-CoA synthetase enzyme. This is done by breaking the thioester bond in Succinyl-CoA and harnessing the energy from that bond in the form of GTP. This reaction is an example of substrate-level phosphorylation, but instead of forming ATP, GTP is formed. ATP can be formed after the cofactors NADH and FADH2 undergo oxidative phosphorylation. In substrate-level phosphorylation, the inorganic phosphate binds to the histidine on the active site of Succinyl-CoA and converts the GDP and inorganic phosphate to GTP. The coenzyme A produced in this reaction was previously used as a coenzyme in the alpha-ketoglutarate dehydrogenase complex in step 4. Overall, delta G of this reaction is negative 2.9 kilojoules per mole. After succinate is successfully formed, the enzyme Citrate Dehydrogenase is used to convert succinate to fumarate. This enzyme is particularly unique because it is the only membrane plasma-bound enzyme in the citric acid cycle. All other enzymes are freely moving in the matrix. In this reaction, succinate is oxidized to fumarate, and FAD is reduced to FADH2 using ubiquinone. This oxidation-reduction reaction forms the carbon-carbon double bond of fumarate with a delta G of 0 kilojoules per mole. The FADH2 then passed its hydrogens to ubiquinone to form UQH2. Since this complex functions off dehydrogenation, which is stereospecific, the trans isomer of fumarate is the only form found. This enzyme can be competitively inhibited by malate because they have similar structures. In step 7, the double bond of fumarate is rehydrated by the addition of an alcohol group to one carbon and a hydrogen group on the opposite carbon of the double bond to form the carbon ion, aka malate. This is another stereospecific reaction, so the trans addition of water to the double bond of fumarate yields L-Malate. The enzyme of this reaction is fumarate hydratase and is a non-hydrolytic cleavage reaction. This reaction has a delta G of negative 3.8 kilojoules per mole. In the final step of the citric acid cycle, L-Malate Dehydrogenase is used to convert our malate back to oxaloacetate to prepare it for the next start of the cycle. This is executed using the reactant NAD+, which means that this is another oxidation-reduction reaction. Malate is oxidized to oxaloacetate, and NAD+ is reduced to NADH. This is a highly endergonic reaction with a delta G of 29.7 kilojoules per mole. This is compensated for in the first step of the cycle, which is the formation of citrate, because it is highly exergonic, so it will pull the reaction through. Overall, the citric acid cycle yields 2 CO2, 3 NADH+, 1 FADH2, or UQH2, and 1 GTP, or ATP. The additional CO2 and NADH+ listed on the animation is derived from the PDH complex. The oxidation of acetyl CoA to CO2 occurs at both steps 3 and 4. The 3 NAD+ are reduced to NADH by dehydrogenase reactions in step 3 with isocitrate dehydrogenase, step 4 alpha-ketoglutarate dehydrogenase, and step 8 malate dehydrogenase. The 1-FAD is reduced to FADH2 in step 6 with the enzyme succinate dehydrogenase. Finally, 1-phosphoanhydride bond is formed in GTP or ATP in the substrate level phosphorylation in step 5 using Succinyl-CoA synthetase. It is generated from the energy stored in the CoA thioester bond. So per pyruvate, there are 4 NADH, 1 from the PDH complex and 3 from the TCA cycle, 1 GTP or ATP, and 1 FADH2. As shown on the left-hand side of the animation, the hydrogen in our NADH+ and FADH2 comes from the reductions that occurred throughout the single cycle. These are conserved in the NADH and FADH2 and stored in the mitochondrial inner membrane until oxidation reactions are necessary, which would form water as the product. These are powerful reactions that assist in the movement of protons from the matrix to the inner mitochondrial membrane space, leading to a gradient that derives the relocation of protons to the matrix, allowing oxidative phosphorylation to occur, thus leading to 3 ATP for every NADH and 2 ATP for every FADH2.